Hello, ma'am. Good evening, ma'am.
Good evening, sir.
Good evening.
You can start now, sir.
Yes, ma'am. Thank you.
Good evening, sir.
Good evening, Gana. Good evening. Good evening, Ajay.
Good evening, sir.
Good evening, Gana. Any doubt in the previous session, my dear students?
No, sir.
Okay. So, last class already, about we were discussed DNA, the structures, and all the important points, isn't it? Today we are going to discuss about the part 2, molecular basis of inheritance, continuity of the molecular base of inheritance. Here you must know DNA as a genetic material, already you know about, but here you have to differentiate according to the organisms, prokaryotic and eukaryotic. What are the differences is there? That things we are going to discuss about in this part. Prokaryon, already you know about. What is by prokaryon? Mean primitive. It is not well developed. Okay, it is not well developed microorganism. What about the nucleus? Nucleus, particularly DNA molecule. It can be invisible manner. It is scattered manner. We cannot able to find out where the exact location. Such type of nucleic acid present in the prokaryotes only. Okay, we may tell as an example, which one you know? So bacteria is an example, prokaryons. Okay, how it will be like that. And eukaryon, well developed nucleus, and also rigid cell wall is also present in it. And also nucleus, inside there is a chromosome and visible manner, just like a thread. Already we know very well about the structure of the nucleus, isn't it? Okay. Here we have to differentiate between these two only. The prokaryote DNA molecule, how it will be, and eukaryotic molecule, how it will be. Okay. How it they constructed? What are the molecule is there? Already we studied the eukaryotic model only. The double stranded DNA molecule is nothing but what is that? Eukaryotic model only. Otherwise, eukaryotic packaging only. See here. So DNA helix in the prokaryotes, how it will be? In term of DNA helix packaging means nothing but the component. What are the component present in it? Okay. The packaging in prokaryote is relatively simpler as compared to the eukaryote, because eukaryote is the highly developed animals, isn't it? Eukaryote, what is that? Developed animal. What about the prokaryote? It is a primitive animals, bacteria, and take an examples of a prokaryote. And higher animals, which one you know? Human beings and plant, as well as a dicot and monocot. The animals, chimpanzee, cow, buffalo, whatever it may be, they are all considered getting known as multicellular eukaryotic nature. So here the comparison between the eukaryote as well as the prokaryote, what are the differences is there? How it will be? Both are similar, but some points they may be differ. Okay, see here. The features of the prokaryote packaging of the DNA helix is as follows. Prokaryotic DNA molecule, what are the packaging material present in it? See, one by one. See here. It's a very theoretical aspects only they have given. See here, in prokaryote bacteria like E. coli. So, the prokaryote means E. coli have taken examples. The size of the DNA is 4.6 million base pair. Note it down, my dear student. Prokaryote DNA molecules packaging. Okay, heading. Followed by example. E. coli is an example. The size of the DNA molecule should be 4.6 million base pair. That is approximately 1.36 millimeter in length. Okay. So when the cut and stretched out, okay, when you are going to cut the, after cut, what happened? When you are going to stretch the DNA molecule, the DNA's total length will be, should be 1.36 millimeter only. Where? In the E. coli. It's a very less number, isn't it? So 1.36 millimeter. It is packed inside the small bacterial cell with a phenomenon called as supercoiling. So supercoiling means nothing but it's a round shape coiling manner, supercoiling manner, which means the DNA is either under wound. Less than 1/10 of the helix per 10 base as it is, or over wound more than 1/10 per 10 base pairs from its normal relaxed state. This is the structure similar to that of the eukaryotic nature, but compared to the base pair. Okay, so this is the another point. Go to the next one. In most prokaryotes, stored genetic material is a single circular chromosomes. Already told you, each and every bacteria, it has a specialized structure. Is that it is nothing but a single standard only. Sometimes it may be a circular standard. Okay, chromosomes located in an area of the cytoplasm and protected only by the outer membrane or the cell wall. Okay, it is not present in the where there is no any definite nucleus. It's present or scattered in the where in the cytoplasm. You know very about the structure of a bacteria. Okay, we cannot able to find where there is a nucleus. Okay, the nucleus, the chromosomes, it may be scattered manner. That's why they consider the getting prokaryote, invisible manner. Okay, see that one. Cytoplasm only is scattered. Okay, the area will be cytoplasm. What will be the nature? Protect only by the outer membrane only. Outer membrane only protect the cytoplasm also. Okay, inside there is a chromosome or DNA molecule present. Okay, such an organism is called as nucleoid. Okay, such an organism are called as nucleoid or genopore. Don't forget, my dear students, which one of the DNA molecules is covered by a cell membrane or cell wall. Such type of what is that organization is called as nucleoid or genopore. Suppose if it is covered by a nucleoplasm, nuclear membrane, it is called as eukaryotic nature. You know about. Okay. Other proteins and enzymes such as the DNA gyrase is another part. Aid in supporting to the supercoiled structure. The double standard structure or single standard structure already you know about. The single standard only present in the prokaryote. Okay, circular nature, covered by a cytoplasm with outer membrane only. Such conditions are termed. That organization is called as what is the nucleoid and also genopore. Not only that, and also some of the protein and the enzymes also what is that supported for the supercoiling structure. So what are the enzymes? You know, DNA gyrase. Sometimes they may ask the questions prokaryotic. Okay, which enzyme is supporting for the maintenance of the supercoiled structure? Like that, they may ask the questions. So I have to tell the DNA gyrase. AS is a suffix for all the enzymes, lipase, protease. Okay, likewise. Okay, then only the structure will be maintained. Otherwise, the structure it will not maintain. Okay, see that. And next one is called as DNA double helix in eukaryote, prokaryote and eukaryote. Here already you know very little about, as it is similar to that of the human being, double helical structure discovered by who? The name of the scientist Watson Crick. Okay, here what they are telling, just listen. The packing of DNA is much more complex compared to prokaryote because it is a highly developed in nature. Okay, then in the prokaryote, that is already know I told. The arrangement of the DNA helix in eukaryote is as follows. What are the arrangement is there? See that one. In eukaryotes, each chromosome consists of a long DNA molecule. There 1.3 millimeter only. Here this is a long nature. Nucleotide. This number of depends upon the length of the nucleotide. Okay, already you know very little about. And have a different types of packing of compress their DNA inside the nucleus. Compressed the nucleus, just see inside it what happened, you know. See that one, so you can easily identify it. Okay, so whiteboard is not here yet. Okay, anyhow, so inside what happened, you know, inside it is compressed. There is a network like that. Just see it, any one of the eukaryotic, there is a nucleus inside is the nuclear membrane, nuclear pore is also there. Inside nucleolus also present in it. Inside the nucleolus, what happened, you know, the chromatin thread also, how many fold is there? We cannot be able to count. Okay, there are so many fold is there. That is compressed. Okay, compressed their DNA inside the nucleus only. This is the first point. Second point, DNA is wrapped around histone protein. Don't forget, my dear students, this is very, very important. Histone protein is also present on it and form a structure called, what is that? U chromosomes. Okay, this is very, very important. Suppose presence of the histone protein on the DNA molecule, that structure is collectively called as nucleosomes. Don't forget, my dear. This is very, very important. So many times they were asked the questions. DNA wrapped around histone protein. It is an actomer, 8 molecule present in a beads-like structure, histone protein. Suppose the DNA molecule contains histone protein, that the entire structure collectively called as what is that? Nucleosomes. Okay, see that one. So prokaryote, the absence of histone protein. The eukaryote, the presence of histone protein. Don't forget, my dear. This is very, very important. Then the DNA is negatively charged due to the presence of phosphate group. Another one is DNA is negatively charged due to the presence of phosphate group. Then only we are calling as a nucleic acid. Okay. Then fourth point, the histone protein that are rich in basic amino acid. This is very, very important. Histone protein that are rich in basic amino acid, mainly lysine and arginine. Note it down, my dear student. This is very, very important. The histone protein composed of 2 main amino acids. That main amino acids called as what is that? Lysine and arginine are organized to form an octamer nature. Octamer. These are the 2 amino acids present in the histone protein. The histone protein is which type, you know? Octamer. Just like a 8 beads close together with the help of the bond. Okay, such type is called as what is that? Octamer nature. Histone protein is also octamer. Mainly there are 2 amino acids that the main amino acid is called as what is that? Lysine and arginine. Don't forget, this is very, very important, my dear student. Okay, okay. So Ajay, the histone protein contain what are the component, my dear?
Sir, nucleosome.
What is that?
Nucleosome.
No, what I am asking, histone protein, isn't it? Histone protein containing 2 amino acids. What are they? What about Ajay? Yeah. Histone protein containing 2 basic amino acids, isn't it?
Yes, sir.
What are they, please, my dear?
Sir, I'm not sure, sir, but is it the H2A, H2B, H3, H4 and all, sir?
No. Are you seeing the slide, my dear?
Yes, sir.
Will you please read the 4th point?
Sir, lysine and arginine, sir.
Yeah, that is very, very important. Now only I'm talking about like this, about this. Dhanalakshmi, me, please, both of you, please follow me, because
Yes,
it
sir.
is very, very important. This part
Yes,
is
sir.
very, yeah. This part is very, very important, my dear students. Histone protein that are rich in the basic amino acid, mainly lysine and arginine. It is in the form of octamer nature. Okay, these are the 4th point only. And also the eukaryotic DNA molecules negatively charged one because the presence of phosphate group. Okay, so this is very, very important. Please follow it, my dear. The octamer is core or nucleosomes core or new body is made up of 2 subunit of, see here, this is very, very important, my dear. H2A, H2B, H3 and H4 histones. There are 2 types of subunit is there. Where? In the octamer core. I will show you the structure. You can easily identify it. Okay. Octamer core or nucleosome core, new body is also made up of 2 subunits. That subunit is called as, what is that, H2A, H2B. What is the meaning of that one? Histone 2A, histone 2B, histone 3, histone 4, like that. The opposite charge of the DNA and histone facilitate the tightly wrapping of DNA around the nucleosome core, that is histone, by the development of electrostatic force between them. Which one DNA as well as the that histone protein molecules. What will be the bond here? That is a electrostatic force. Then only it is bind on the DNA molecule. The histone protein will bind on the DNA molecule in octamer nature. It has a 2 subunit. What are the subunit? H2A, H2B, H3 and H4. Okay, so this is very, very much. These 2 points is very, very important. And then about 200 base pair of DNA is wrapped over new body to complete about 134 tons. See here, about 200 base, but these are all are not comes into the prokaryote DNA molecule. That's why it is a complicated one, because prokaryote is a simple one only. Less number, visible nature, invisible or visible, invisible nature. It is surrounded by the only cytoplasm, but eukaryote, it's present in the nucleus in thread-like structure. Many fold is also there, and it is made up of histone protein. The histone protein is also has, what is that, 4 subunit. What are the subunit is there? In the DNA and the histone, there is a attraction between each of them with the help of electrostatic force. And also, how many base pair is present in it? 2,200 base pair of DNA molecule, and each and every 134 times. This forms a nucleosome size 110 into 60 Armstrong. Note it down, my dear. This is very, very important. And eukaryotic, the nucleosome size should be 110 into 60 Armstrong. Okay, this is also a important point in this part. See, and then 8th one, the adjacent nucleosome are bound to each other with the help of the linker DNA having length of about 80 base pair. Each and 80 base pair, there is a linkage is also present in it. This is the 8th point. The 9th point, the linker DNA is held in a place by the H1 histone. Which one is the linkage? Like H1 histone protein is the linkage. 10th point, the nucleosome undergoes stacking over each other to form a solenoid or chromatin fiber. Nucleosome is also undergo stacking over each other to form a solenoid or chromatin fiber. These are the points is very, very important. Solenoid, okay, chromatin fiber. It is a hair-like structure. Okay, see, these are the important. And also having 14, 15, 16, 17. This is the considered as a histone protein. Nucleosome structure as given here. So here, this is the DNA molecule. The round is there, this is the DNA molecules. Wrapped the histone protein molecule. This is histone protein. Okay. Histone protein, collectively called as what is a core of histone molecule. This is collectively called as core of histone molecules. Collectively called as core of histone molecule. What are the histone molecules? Core of histone molecules mean DNA molecule present in it. Histone octamer. This is the 8 beads present in a single structure. And followed by H1 histone is also present. They are collectively called as what is a nucleosome and also called as core of histone molecules. Okay, this is the structure. This is very, very important, my dear students. Don't forget. See here. The stacking and further coiling is supported by non-histone proteins. Some of them are having a linear structure. Okay, there is a beads, just like a what is that? Gap in between. There is a gap is there. But without a histone protein, that is non-histone protein molecules also present in between the histone molecules, histone octamer. Okay, see. See this? So whiteboard means it's better for your understanding. So see the structure. Take an example of this is a DNA molecule. DNA single strand. Okay. The single strand, it contains a, what is it? Octamer nature. Octamer means, you know about the, this is called as octamer. This is called as octamer. Okay. So this one is, what is that? DNA stand, isn't it? DNA stand. What about this one? This is considered as named as a octamer. Which one you know? Histone protein. Histone protein molecules. This one is histone protein. In between there is a space, isn't it? In between there is a space. This is called as what is a non-histone protein. Non-histone protein. Okay, this is the structure, but already we saw it, no? So that there is a round shape is there. It is covered by a which one, DNA molecule. DNA molecules covered. This is called as octamer in nature. Octamer means, just imagine the structure will be like this: 1, 2, 3, and 4, 5, 6, and 7, and 8. These are the octamer structure. What are the amino acids present in this area? Already you know very about. So, okay. So what are the things you know? So one is called as arginine. Arginine. Okay. This is called as arginine. Another one is
lysine.
Yes, lysine. Very, very important. Okay. So this is lysine. Lycine. Okay. So these are the things. But this is considered a which one? Octamer in nature. This is surrounded by the DNA molecule. This is surrounded by a DNA molecule and followed by there is a core. Is that which one? Histone protein core. Okay. So this part is called as what is that? Histone 1. Histone 1 molecules. So this is the DNA molecule octamer, histone protein molecule. This is a DNA molecule. This is a histone protein 1. Okay, so this has given in your book. Okay, my dear. Are you clear? Ajay?
Yes, sir.
Okay, this is considered a chain, a single polypeptide chain. Take an example. So this part, this part is not mentioned any protein molecule. That's why they given us a non-histone protein area. This one is protein area, just like a beads. This beads is called as what is known histone protein. It has a octamer nature. Octamer. Okay, octamer. What is the meaning of octamer? Means so 8 molecules present. 8 molecules in a beads. So there is a space, just like there is an interval between the two beads also there. That condition is called as non-histone, but that portion is called as non-histone. This is histone protein. The histone protein structure already mentioned here. Okay, it composed of these are the 2 amino acids. Very, very important thing. Okay, okay. See that one. Okay. Okay. So next one is what is the name? The stacking of further coiling is supported by non-histone protein. The each helix of solenoid consists of the approximately 6 nucleosome. Each and every helical structure it contains. How many nucleosome is there? 6 nucleosome is there. I mentioned here 5 beads is there, no? So 6 nucleosomes stacked over the each other. This causes the length of the solenoid to become around 13 nanometer, 30 nanometer into 50, like that you have to multiply, we'll get it to 35. Sorry. There are mainly 2 types of chromatin fibers. Is there? One is called as euchromatin and another one is called as heterochromatin. In nucleosome, it contains 2 types of, what is that? Chromatin molecule. That chromatin molecule is called as what is known? Euchromatin and heterochromatin. Okay, so these are the important. See that. The euchromatin fibers are 30 to 80 nanometer. Note it down, my dear. Nucleosome is contained histone protein, non-histone protein part also there. The octamer nature, lysine is a protein molecule, lysine is amino acids, and arginine is also amino acids. Okay, and then it contains 2 types of chromatin is there. The 2 types of protein, one is called as euchromatin, and another one is called as heterochromatin. The euchromatin fibers are 30 to 80 nanometer in diameter and loosely packed and stain light. That is very, very important. Euchromatin, when you are going to stain, what happened? It is a very light color in nature, loosely packed. That's why it is called as euchromatin. These fibers make up about 2% of the genome and mainly consist of genes. How many percentage only? Out of 100, only 2% is euchromatin. Okay, so here you must note it on 3 points only. Structure will be 30 to 80 nanometer. Okay, loosely packed, right? And third one, in 2% of the genome mainly consists of genes. 2% only present in the genome. And next one is called as heterochromatin. It's very, very important one. Fibers are about 300 nanometer in diameter and are more densely packed. Here loosely packed, here densely packed. And stain is dark. It's look like a dark manner. These fibers make up about 98% of the genome and mainly consist of repetitive DNA. Don't forget, my dear. Sometimes they may ask the question, the repetitive DNA molecule mostly found in which one of the following chromatin? Euchromatin or heterochromatin or some other pseudochromatin, like that. They may ask the questions. And also, how many percentage of the gene is occupied? That is also another question. 98% of the genome mostly present the heterochromatin is nature. What about the diameter? Diameter will be 300 nanometer. What about the nature? Stain dark in nature. It's not light nature. It is a dark in nature. Okay. So these are the difference between euchromatin and heterochromatin. Okay. Next one is the solenoid undergoes further condensation during the metaphase stage to form a chromosome. Don't forget, my dear. Already we studied anaphase, prophase, anaphase, metaphase, telophase, isn't it? In prophase stage, what happened? the condensation takes place. The simple thread-like structure. Simple thread-like structure is the simple thread-like structures also condensation. After the condensation, it forms as a chromosome-like structures. I will show it. It is better for your understanding. Okay, see that one. So basic and whiteboard. See there, and then have to delete all those things. See, my dear students. Examples, any one of them. Okay. Wait a minute, my dear students. So it is not moving. Okay, Ajay. See here, it's not coming. So once again, I will try. So it can better for you. Okay. See that, my dear students. Take an examples. So this is considered a. Just imagine this is a. Imagine as a cell, isn't it? It is considered a cell. Okay. Just imagine. Okay. Just. So inside there is a nucleus, is there, isn't it? Inside there is a nucleus. Inside there is a nucleus. See here, you know the difference. Then only it will be better for you. So this portion is called as what is now nucleus. Okay, this is nucleus. This one is called as what is a cell membrane or cell wall. Okay, inside what happened, you know, nucleus is that during the cell division condition, what happened, you know, the structure will be like this. The structure will be like this. There is a small thread-like structure only. There is a small thread-like structure. This is called as chromatin thread. Okay, during the prophase condition. When it condensation, when it undergoes the condensation process, how it will be changed, you know. See, this structure will be changed into, see here. So this is the structure. How it will be changed? See, the structure will be like this. This is called as condensation. Okay. Just to saw a small rough diagram only, just imagine. So look like this one only. Okay, this structure will be condensed to the formation of the this type when, you know, during the metaphase. Okay, during metaphase only, these changes occurs. That is told here. Inside there is a DNA molecule. You know about the DNA molecules, and the gene is also present inside. It is also two types is there. One is called as what is that? Euchromatin and heterochromatin. Okay, the heterochromatin. Okay, euchromatin, how many percentage is there now? You know about the 2% only. But at the same time, how it will be packed, you know, this is loosely packed. This one is loosely packed. Heterochromatin is 98%. 98% of the genome present in it. But also, how it will be packed? Closely packed. So this is very, very important. Okay, loosely packed means just imagine this one. See, just like this one. So this is loosely packed. Okay, likewise. Suppose if it is a closely packed means, what is the meaning of closely means? You have to put like this. Okay, so this is closely packed. This one is loosely packed. You know the difference between these two. Okay, my dear. So this is very, very important in this category. Okay. See that one. Okay. So heterochromatin, already we studied these things. This metaphase stage to form a chromosome. The structure already I mentioned, isn't it? Loosely packed solenoid undergoes further condensation during the metaphase stage to form a chromosomes. Okay. So this is already we saw it. This is the structure of the what is the nucleosome structures. This present in the octamer nature. Already we know it, but the DNA molecule surrounding the histone molecule is octamer nature. 8 molecule present inside, and also this one is also H1 histone protein molecules. It is a maintain their structure of the beads, collectively called as core of histone molecules. The entire structure is collectively called as what is the nucleosomes. Okay, this is very, very important, my dear students. This can be varied. So this one is also another type. There's a 4 subunit. Already we saw it, 4 subunit is there. H2A, H2B, H3, H4. These are the 4 subunit. This is histone. What is that? H1 protein. Then only it tightly packed. Suppose the histone protein is not here, it can be used loosely. What is it? Covered. That is a very, very important one. So this is the DNA molecule. Covered a octamer of histone octamer. The inside there is a subunit is the, okay, these are all at present where, prokaryote or eukaryote. This is mostly present in the highly developed eukaryotic nature. Then only they are telling us that it is a very complicated manner. Okay, my dear. Prokaryote is not like this. There is no histone protein and linear manner only, simple molecules only. But in this category, well-developed nucleus is also present in it, and chromosome is also well-developed. And inside there is a DNA molecule. Is the DNA molecules? What is that? Histone protein is present in it. Then only it is highly developed in nature. The histone molecules inside, what are the structure is that? These are the things. Without these things, what is that? The DNA structure is not maintained. Okay, so this is the structure. See that one. So these are all, each one there is a bead is there, long beads. These are all are called as what is known now? Histone protein, octamer in nature. In between the 2 histomer, there is a gap is there, isn't it? There is a linker. Okay, this is the linker. So these molecules as well as these molecules also link with the linker of the DNA. The linker of the DNA on it, what happened? There is a non-histone protein. That's why I told you. This is histone protein. This is non-histone protein present in it. So this is the entire structure of the DNA molecule. Any doubt, my dear students, in this part?
No,
Ajay?
sir. No doubt, sir. No, sir.
Okay, carry on. Okay. What about Dhanalakshmi?
No, sir.
Okay, see here. So go to the next one. So this is the question. See that one? Histone proteins are what is that? Whether it is a basic, negatively charged, basic, positively charged, acidic and positively charged, and also acidic, negatively charged. See here, nucleosome is there. Here 8 protein mark, that is called as proteins, histone protein. Wrap DNA, there are 2 types. Wrap DNA is a acidic in nature and also negative. So that is neutralized by the positive component. So, sir, what is that component here? What is the examples? This is the question for you. What is that? Basic component and also positively charged. This is the result. Basic and positively charged histone protein molecules are basic in nature and positively charged. That is very, very important. And suppose if it is negative, what happened? Neutralize the positive charge. This is the combination. Okay. So don't forget, my dear, this is the questions. Okay. Difference between the packing of DNA helix in the eukaryote and prokaryote. Already we studied that things. It may be given in the tabulation format. Prokaryote and eukaryote. Prokaryote DNA is found in the cytoplasm. Don't forget, my dear. Prokaryotic DNA present in the cytoplasm. Here, eukaryote DNA packed into the nucleus of the cells. Some DNA found inside the chloroplast and mitochondria. This is called as Extra chromosomal mitochondria, already we studied in the previous chapter. Okay. Introns are absent. Introns are absent. Don't forget, my dear. Introns, in between, there is a molecule is absent. The introns are present. It contains a small number of genes and is organized into single chromosome only present in it. Number of genes also present. But here, it contains a large number of genes and it is organized into many chromosomes. See here, it is a circular and not bind with histone protein. It is a circular nature. Prokaryotic maximum, it is a round, circular nature, but helicals. But here, it is a linear with two sides and packed with histone protein molecule. This is very, very important. The genetic material packing is referred to as a nucleoid or genophore. Here, the prokaryotic, what is that? Genetic material packaging is referred to as the nucleoid and genophore. But in this category, there is a chromatin is there. That chromatin is also different into euchromatin and heterochromatin. Here, nucleosomes are not present, but here the nucleosomes are present. What is the meaning of nucleosome? The octamer nature, completely entire structure of the histone protein, octamers, and also the non-histone protein, isn't it? Entire structure is called as nucleosome core or histone core, like that. But in this category, it's not present, but it is present indeed. Okay, so this is very, very important, my dear student. You have to note it down in this part, the nucleoid and genophore, chromatin structures. Where it present, it is a nucleus, it is a cytoplasm. Okay, which one is absent here? Intron is absent in the prokaryote, intron is present in the eukaryote. This is very, very important, my dear. Okay, take a notes, then only it will be better. And one more is there. There are two types of protein molecules have been reported. One is called as histone protein, another one is called as non-histone protein. How they are differentiated between these two? See here, histone, basic globular protein, globules, just like a round. Okay, here structural, enzymatic, and regulatory role is also done by the non-histone protein. Highly conserved protein. Okay, rich are arginine and lysine amino acid. This is very, very important. This is very, very important. In this category, acidic protein rich in tyrosine and tryptophan. These are all aromatic, isn't it? Tyrosine, tryptophan. These are all aromatic amino acid present in the non-histone complex. But histone protein only contain lysine and amino acid. This is very, very important, my dear students. So many times they were asked the question. Histone is a low molecular weight, but non-histone is a high molecular weight. Synthesis occurs during the S phase, where there is a cell cycle. G1 phase, S phase, G2 phase, M phase, and G0 phase, isn't it? So you have to note it down. S phase is very, very synthesis means DNA synthesis takes place. Synthesis phase inside cycles. This is synthesized throughout the cell cycle. This is a non-histone protein throughout the cycles. G1 phase, G2 phase, S phase, M phase, likewise. Absent in prokaryote. This is very, very important. Histone protein completely absent in prokaryote. But non-histone stimulate genetic activity. This is extra points. Act as gene repressor in non-specific manner. Gene repressor. Repressor. Okay. This is another part. The non-histone proteins include DNA, topoisomerase enzymes, RNA polymerase. These are the enzymes. What is that? Activate. The non-histone protein included DNA topoisomerase and RNA polymerase. Both are involved in the replication process, semi-conservative method. Okay. So don't forget, my dear. This is very, very important. You know the difference between, what is that? Prokaryotic DNA and eukaryotic DNA. And also, you know the difference between the histone protein and non-histone protein. So this is very, very important. Okay. So this is the structures. See that? H3, H4, H2A, H2B. These are the structure is there. This is dimer. So this is also a dimer. So combine contact close together as a result, you will get it the octamer nature. This is the tetramer nature. So these are all united together form as a tetra nature. When the tetramer, here also the tetra is joined together, you will get the octamer. The octamer present in the center, surrounded by the DNA molecules. Okay, so don't forget, my dear. This is a structure. They have given a single structure, isn't it? 4 subunit is there. 1, 2, 3, 4. But in this category, 2 is present here. See, dimer nature, this one, dimer. Again, the dimer converted into tetramer, because addition of the 2 dimer, you will get tetramer nature. Likewise, these 2 molecules, H2A and H2B also, united together dimer, and form as a tetramer. When it addition of the another dimer, you will get the tetramer. So tetramer, here also tetramer joined together as a form of octamer. 4 plus 4, you will get 8. Okay, it present in the octamer in nature in histone protein molecule with DNA. Okay, I think there is no doubt, I think, isn't it, in this portion. So these are the different. Okay, go ahead. So this is called as the DNA structures. This is nucleosomes. See here, this is the structure of nucleosomes. Each round is considered a histone protein. Entire structure is called as nucleosome. Okay, this is called as chromatin. This is called as complete. How many octamers present inside? See that contains it, condensation with that. Okay, this is called as chromatin. This is chromatin loops. This is chromatin loops. Condensation means more than one united together, one above the other. Condensed chromatin loops. Okay, finally you will get chromosomes. See that nanometer differences is there. 2 nanometer, already we studied, isn't it? 11 nanometer, 13 nanometer, 300 nanometer, 700 nanometer. It's already double here, 1400 nanometer. You will get it, chromosome, from where you know, in the nucleosome to chromosome. This is the condensation. Condensation means concentration, increase the concentration. Addition of more amount, more number of, what is that? Chromatin loops or chromatin thread on it. As a result, finally what happened? No, converted into chromosomes. Okay, don't forget, my dear. Okay. Search to identify the genetic materials. Another one is, how will you prove the DNA as a genetic material? Will you please? Any ideas? Any methodology? Any experimental present in it? Already known one, please. Let me share. Ajay?
Yes, sir.
Do you know DNA as a genetic material? How will you prove? Any methodology?
So, no, sir, not sure, sir.
Okay, okay. Anyway, what about Dhanalakshmi, please?
It's a Griffith's experiment.
Very good. Griffith's experiment. Very good. Ajay, note it down. Okay.
To mice, they'll inject, sir.
Yeah. What are the strain is present in it? Will you please?
S strain and R strain, sir.
What about S strain? It's denoted as?
Smooth, sir. R
Okay.
is RF strain, sir.
Okay. You know the concept, isn't it?
S a little.
Little. Okay. Just to go through it, you will get the idea. Okay. How it will be possible. I will show you, but here also it's coming. Okay. No need to worry. Okay. Thank you. Okay, Ajay. Okay. Follow, please. Genetic material. Okay. Genetic material can transfer bacteria. A British doctor, S.F. Griffith, first studied this transformation in 1928. Note it down, my dear student. In the year, very, very important. 1928, Griffith experiment observed one of the microorganisms is called as Streptococcus pneumoniae. Okay, the popular known as pneumococcus. Don't forget, my dear. This is very, very important. 3 points here. Genetic material can be transferred bacteria. Yes, DNA is also transferred from the one bacteria to another bacteria through the way of conjugation canal or whatever it may be. So many things. When in common contact close together, there is a bridge in between the 2 bacteria. So one bacteria transfer the genetic material into the another one. Okay. After that transformation, what happened? It will be split into 2 halves. It will be split. Okay. So this is the methodology and also the reproduction process. Conjugation tubes also formed. Already you know about that things also. So this was done by the name of the scientist S.F. Griffith in the year 1928 with the help of the microorganism is called as what is a Streptococcus pneumoniae, popularly known as pneumococcus. What he did? Okay, already see that this is the experiment. Can you see the slide, my dear?
Yes, sir.
Okay, this is
Yes,
the
sir.
experiment. Yes, Ajay, just okay. See here, what he did, the grip experiment, also done by the bacteria is called a Streptococcus pneumoniae. This is very, very important. Which cosmetic organism used by the Griffith and also proved DNA as a genetic material. This is very, very important, my dear. Okay, he has chosen two strain. This bacteria causes pneumonia. Okay, two strain of this bacteria were used. One is called as rough, they denoted as R strain. Another one is called as S strain, that is called as smooth strain. Who did Griffith? Which bacteria? Streptococcus pneumoniae. Okay, two strain is there. One is called as R strain, another one is called as S strain. See that R strain is also called as, note it down, my dear, smooth mucus polysaccharide coat. Don't forget. Rough strain means it is not a smooth one, opposite to the smooth. Resistant to immune system. Note it down. This is also very, very important. Yes. Strain. Smooth mucus polysaccharide coat. The strain is covered by a smooth. There is a jelly-like structure. That's why they have told us a smooth mucus polysaccharide. But it is also very important for the resistant functions, resistant to immune system. The virulent is a R strain. Lack the coat. The coat is not present. The smooth mucus polysaccharide coat is completely absent here. But at the same time, what happened? Destroyed by immune system of the host and non-virulent. Okay, so this is a important things. See that one? This is the experiment. What the Griffith experiment here explain, he chosen a mouse or mouse. Okay, he injected the strain, rough strain, injected directly. What happened, the mouse will be alive in nature. At the same time, smooth strain is also injected into the mouse. See that this is the smooth strain. The round is the, what is that? Smooth in nature, the mucopolysaccharide coat. What about here? Rough means there is a small needle-like structure. See the difference between these two strains. Okay, the outer membrane cover. Okay, there is a needle-like structure. Okay, the shape of the strain. This is the smooth one. When the smooth strain is injected to the mouse, what happens? It dies. We come to the conclusion. When we are injecting the smooth strain, why the mouse dies? After that, what happened? Heat killed smooth strains. This one, heat killed smooth strain is a injector. And what happened? The mouse is alive, lives in nature. Then after that, mixed with these two strain. See here, rough strain and a heat killed smooth strain. These two only. This one as well as the first one. These two. That time what happened? The mouse is die. What will be the statement here, my dear? Dhanalakshmi, what happened here?
Sir, I think rough strain will die, sir, and S strain will remain, sir.
No. Okay, your point of view, rough strain and heat killed smooth strain. Okay, heat killed means what happened here?
So, in heat killed means that the strain is dead, sir. It won't
Okay.
have any effect, sir.
Yes, good, super. So what happened in the last experiment? Last one.
Sir, they are injecting both the normal, sir, rough strain as well as the heat killed smooth strain.
Okay. The genetic material transfer where to where? Normally, smooth DNA molecule is there, Smooth, isn't
Yes,
it?
sir.
What about rough strain? Rough strain is not, and heat killed one. Okay, rough strain and heat killed smooth only. Smooth only heat killed, isn't it? The
Yes,
rough strain, they are having the DNA molecule. It transfer into where? The smooth molecules. Then only the result will be die. Are you clear in this part? Once again, repeat for you. In
Yes,
the last part, listen. There are two what is that? Strain is also present. It's rough as well as the smooth, isn't it? But smooth is killed one, isn't it? Killed means, see the result, killed one, what happened? No, the mouse is alive, isn't it? But rough strain, it is not killed, isn't it? Here the DNA molecule is also transferred into the heat killed one. The DNA molecule is here. It killed means what is the DNA molecule transfer from the rough strain to smooth strain. So the smooth strain effect will be expressed. That is called as genetic transformation. Okay, are you clear now?
Yes, sir.
You
Yes,
have
sir.
any doubt in this part?
No, sir. No
No,
doubt,
sir.
sir.
Once again, I repeat for you. Once again, because it's very, very important, my dear. When your rough strain is injected, the mouse is live. Smooth strain injected, what happened? No, the mouse is die. This is basic. After the smooth strain was heat and killed, the mouse is alive. What they are doing, the rough strain, it is not heat killed. Note it down, my dear. Rough strain plus heat killed smooth strain injected means what happened, the rough strain DNA molecule transfer into smooth, what is that? Killed DNA molecule. So the smooth DNA effect, strain effect will be expressed in the result. You got it now?
Yes, sir.
Okay, good. That is called as a gene transformation. This is the proof of DNA as a genetic material. Okay, because the effect of the smooth strain will be effect here. Okay, they will produce which one? Similar effect like I say a smooth strain. That is the reason this was discovered by the scientists. The same effect here. See that rough, non-virulent. This is virulent. Smooth one dies. So heat killed means it is alive. When both are injected, so its mouse is also die because the live type they expect in blood samples from the dead mouse. Okay, this is very, very important. So we will finish it up to the this level because the time is up. This virulent strain causes pneumonia. These bacteria are known as S-type because when they grow on a suitable medium, they form a smooth colonies. Okay, the outermost layer, just like a structure, you can see it a smooth nature surrounding the cell wall. So it is called a smooth colonies. The non-virulent type of bacteria does not produce the diseases. They are therefore called rough or R-type. Rough, the small colonies. Already we know the structure, isn't it? Griff could kill the bacteria by heating them. He observed that the heat killed S-strain bacteria injected into the mice or mouse did not kill the mice. After the heat killed, what happened? No, it will not produce the effect. It will not produce any effect. But when he injected the mixture of heat killed S-strain and live or bacteria, the mice died because he concluded that some substances needed to produce the mucus coat were passed from the dead bacteria S-strain to the live one of R-strain. That is, the become transferred. This is the major content of the experiment. Okay, he concluded that some substances need to produce the mucus coat were passed from the dead bacteria of S-strain, smooth strain, to the live one of the R-strain. That is, they become transferred. Although Griff did not understand the nature of the transformation, he theorized that some substances in the polysaccharide coat of the dead bacteria might be responsible. Okay, he called these substances the transformation principle. Transformation principle, like that he told. The genetic material of the DNA, we will see the next classes. What we discussed in this part, see that one. Okay, this is the double helical structures. What is that? Second part, already know about. So DNA helix in the prokaryote, what will be the nature? And eukaryote. Okay, there is an enzyme. No, don't forget, my dear. Prokaryote, there is a DNA high-rise enzyme is also very, very important. And circular manner, single standard, surrounded by the cell membrane. Okay, and then DNA double helical structure is a complex manner because what are the things is present in it? Histone protein, tightly packed. Okay. So not only that, and also structure is also vary, and histone protein also the negatively charged phosphate molecule, because the presence of phosphate molecule. The octamer is a lysine and arginine. Okay. So that thing is also very, very important. What are the 4 subunits? Don't forget, my dear, it's also alternative form of they may give and also ask the questions. NEET point H2A, H2B, H3, and H4. Sometimes they may give H2A and H2B. Okay, H2, 3, H2B3, H2BA2. Like that, they may shuffled. Okay, confused you. Don't forget. This is very, very much. These are the 2 subunits: H2A, H2B, H3, H4. That's all. Okay, 4 subunits there. And histone protein is the electrostatic force. Then only it will be maintained their structure. And how many base pair is that? Don't forget, my dear student. 134 tan is there. 110 into 60 Armstrong is there. And also 80 base pair of the length. H1 protein is very, very important. And solenoid and chromatin fibers, it's a thin like structures. Non-histone protein parts also present in between the 2 solenoid. How many nucleosome present in it? No, each and every helical structure, there is a 6 nucleosome is also stacked over each other. 39 amino. Okay. And then there are 2 types of chromatin molecules also present in it. One is called as euchromatin and heterochromatin. So euchromatin, what is that? How many percentage? Heterochromatin, how many percentage? Okay. When it happens, metaphase stage. Okay. This is very, very important, my dear student. This is the structure of nucleosomes. Okay. Remaining things we will see the next classes, my dear students. Please go through it. You will get the ideas. Okay. Thank you so much for the participation.
Thank you, sir.
Thank you, Pa. Thank you, Ajay.
Thank you,
Thank you.
sir.
Thank you, ma'am.
Thank you, sir.
Thank you, ma'am. Thank you so much.